I'll be talking about the effect of temperature on enzyme function. So starting off with what is an enzyme is a biological catalyst, which means that it's a substance derived from a living organism that acts as a catalyst to bring on a specific chemical reaction. And catalysts are used to initiate the lowering of activation energy so that the enzymatic reaction rate can speed up. Enzymes bind to substrates at activation sites, so substrates can undergo a chemical change and begin their reaction, in which the enzyme isn't consumed in the reaction. Changes in activation sites don't allow substrates to bind, resulting in no reaction. The substrate used in this experiment was azocasein, and azocasein is a milk protein combined with dye that can be broken down by trypsin. And enzymes are important components of life because they help with the process of digestion and metabolism. And the enzyme that we used in our lab was trypsin. And trypsin is a protease enzyme that is in animals, bacteria, and humans. In bacteria, trypsin is found in the cells. In animals, the bloodstream. And in humans, the inactive form of trypsin is synthesized in the pancreas and is used for digestion of dietary proteins found in the small intestine. And trypsin catalyzes the hydrolysis of peptide bonds by breaking proteins down to smaller peptides. And the peptides are then further hydrolyzed as amino acids available for absorption from our bloodstream. Our experimental design was based on our prior knowledge of enzymatic functions. Therefore, we knew that any change in environment could alter the shape of the enzyme or the activation site. The conditions that were most common were temperature, pH, and salt concentration. So in our experiment titled Effects on Enzyme Function, we wanted to know what would happen to trypsin if we increased temperature. Therefore, the condition chosen was temperature, and it was hypothesized that as temperature increased, enzyme function would also increase to a certain extent. The rationale behind that was that as temperature increases, the particles in the solution speed up and spread out, resulting in the overall reaction rate of the enzyme function being sped up. And then, as you can see, the sources are listed. And then for materials, I'll go step by step. We had to prep and label microcentrifuge tubes. Then for each experimental treatment, we added the following to a clean centrifuge tube, which was 450 milliliters of 0.15 molar NaCl and 20 nanometers of HCl pH 8, 500 milliliters of azocasein, 50 milliliters of trypsin, and then no trypsin for the controls. Then we incubated for 20 minutes at the 4 different temperatures, which were 15, 28, 35, and 50 degrees Celsius, having 3 experimental replicates for each temperature and 3 controls for each temperature. And we added 500 microliters of 10% TCA once the time is completed. Place each tube in the centrifuge for 2 minutes at 12,000 RPM. Transfer supernatant into clean cuvette, blank the spectrometer at 440 nanometers, and read the absorbance in the spectrophotometer at 440 nanometers and record the results. And then behind some of the reasons we did that, first of all, the experimental variable was temperature, and the controls that were used were the specific conditions of the control variables. So that would be 450 milliliters of 0.15 molar NaCl, and then the 20 nanometers of the pH 5. And then we had 3 negative controls, but no trypsin for the negative controls. And then we had 3 replicates at each condition. And then we added TCA in order to stop the reaction and centrifuged it because we needed to see the difference. And then getting to results. As you can see, the results are listed on the chart, and the absorbance of enzyme activity at 50 degrees Celsius was significantly greater than the absorbance at 35 degrees Celsius, as you can see with the p-value being 0.063, and then the enzyme activity increased by 0.304 as the temperature increased by 15 degrees. And by enzyme activity at 0.304, that means the absorbance And the absorbance of the enzyme activity at 35 degrees Celsius was not significantly greater than the absorbance at 28 degrees Celsius. And then the enzyme activity increased by 0.035 as the temperature increased by 7 degrees Celsius. And then the variability of all trials, which was the standard deviation, was high for the majority of the trials. And then going on to discussion. It was hypothesized that an increase in temperature will cause an increase in enzyme activity up until 35 degrees Celsius. Above 35 degrees Celsius, enzyme activity will decrease. The results don't support this hypothesis because Table 1 shows the absorbance at 35 degrees Celsius is significantly different than the absorbance at 50 degrees Celsius with a p-value of 0.063, and the absorbance at 50 degrees Celsius increased by a fold of approximately 2.77 from the absorbance at 35 degrees Celsius. Table 1 also shows that the absorbance at 28 degrees Celsius is not significantly different from the absorbance at 35 with a p-value of 0.526. And the pH of 9 and salinity of 0.15 molar at which the trypsin was tested did yield the maximum activity. Work by both groups C and E suggests that the optimal pH for trypsin function is a pH of 9, and work by group F suggested that the optimal salinity for trypsin function is between 0.0 and 0.6 molar. And these findings suggest that trypsin may provide successful advances in bacterial biology when incubated in optimal temperature with a pH of 9 and salinity of 0.15 molar. And we found that the experimental design was adequate to test the hypothesis because the independent variable temperature is clearly defined, the dependent variable absorbance is also clearly defined, and then the control variables, pH and salinity, are also clearly defined. Both the experimental and control groups were tested at various temperatures with 3 trials at each temperature. The results of each trial were measured and recorded using a spectrophotometer, and the results were summarized by using experimental averages, control averages, normalized averages, standard deviations, and p-values. And then the normalized averages as well as the standard deviations were then graphed in a way that related back to the hypothesis. And Group B's experiment showed enzyme activity increasing as temperature increased with an optimal temperature of 60 degrees Celsius, then a decrease in enzyme activity as temperature increases past 60 degrees. And some sources that contributed to variability between this could be fingerprints during spectrophotometry and incorrect micropipetting. We observed that fingerprints can contribute greatly to incorrect results. We also observed that errors regarding micropipetting can yield incorrect results, but only to a certain extent. Our second design was much more adequate than our first due to the addition of more experimental and control trials. The design can thus be further improved by adding even more trials in order to get more accurate averages, as well as testing more temperatures in order to more accurately pinpoint the peak enzyme function.